Hello, I'm Takayama from Kyoto University. Today, I'm going to talk about our COVID-19 research project using iPS cells and organoids. First of all, I'd like to thank AMS Bio, Ajinomoto, Matrixome, and Xenoac for giving me such a great opportunity. And I would like to thank the ISSCR staffs for preparing the virtual annual meeting during the COVID-19 disaster. And first, I would like to make a brief self-introduction. I was at Osaka University before coming to Kyoto University. And at Osaka University, I was conducting research on the differentiation of human ES and iPS cells into hepatocytes and also intestinal epithelial cells. Our human iPS cell-derived hepatocytes have been marketed by Repocell since 2012, and our iPS cell-derived intestinal epithelial cells have been marketed by Takayama Bio since 2019. And at Osaka University, I was also developing a photoactivatable genome editing system. Currently, our laboratory is working on 3 topics: COVID-19, organ chip, and optogenetic projects. I started the laboratory at CiRA on March 1st at 2020. This is the same time as the beginning of the COVID-19 pandemic. I used many viruses before, so I started the COVID-19 project. We are now conducting experiment using SARS-CoV-2 in the BSA facility or Virus Institute in Kyoto University. I do not have enough time today, so I will only introduce our COVID-19 project. We have many cells and SARS-CoV-2 strains for COVID-19 research in my lab. In addition to cell lines such as Vero cells, CAL3, and CACO2 cells, we are conducting SARS-CoV-2 infection experiment using organoid and primary cells. We use many SARS-CoV-2 variants, including original EU and UK and Brazil variants. Currently, most COVID-19 patients in Japan are infected with UK variant, which is known as B1.1.7, so we are mainly using the UK variant. We are also isolating SARS-CoV-2 from COVID-19 patients in our lab. We are now evaluating many drug candidates, such as small molecule compounds, cytokines, siRNA, peptides, and antibodies. Most of these are collaborative studies using our cells and viruses to examine their antiviral effect. We are now collaborating with more than 20 universities and companies. And please feel free to contact us if you'd like to develop a COVID-19 therapeutic agents with us. We have already published 10 COVID-19 papers, and we are interested not only in developing cell model but also in the functional analysis of SARS-CoV-2 and their proteins. As many of you may already know from TV or newspapers, I would like to introduce the background of COVID-19 first. The first COVID-19 patient was reported in Wuhan in China. And in February of 2020, ICTV named this new coronavirus as SARS-CoV-2, and WHO names this disease as COVID-19. In March 2020, WHO declared the COVID-19 pandemic. My laboratory also started in March in 2020, and I started SARS-CoV-2 research because I had a lot of experience in virus experiments. And as shown in red, the number of COVID-19 patients is about 0.16 billion, and the death toll is about 3.4 million. The number of COVID-19 patients and deaths is approximately 10,000 times higher than the SARS and MERS. Although the number of COVID-19 patients and deaths continue to grow, but no effective treatment has yet been developed. The development of COVID-19 therapeutic drugs has been delayed compared to the COVID-19 vaccine. Although remdesivir and plitidepsin are effective in some COVID-19 patients, but it is desired to develop a COVID-19 therapeutic drugs with much higher therapeutic effect. To develop a therapeutic drugs, an excellent model for drug discovery is necessary. This slide describes how SARS-CoV-2 enters the cells. First, SARS-CoV-2 binds to the receptor ACE2, and it is known that some mutation in the spike protein increase the binding force between spike protein and ACE2 protein. Next, the protease TMPRSS2 cleaves the spike protein and split into S1 and S2 domains. And finally, SARS-CoV-2 uses the ACE2 domain to fuse the viral membrane with the cell membrane. Therefore, SARS-CoV-2 infection is highly dependent on the expression of ACE2 and TMPRSS2 in human cells. However, because several important receptors and proteases are known in addition to ACE2 and TMPRSS2, but SARS-CoV-2 infection efficiency is determined by more than two factors. There are many in vitro and animal models that can be used in SARS-CoV-2 research. Vero cells, which are commonly used in SARS-CoV-2 studies, can efficiently replicate SARS-CoV-2, but these cells are African green monkey kidney cells, not human cells. Therefore, we consider that it is necessary to use a model that can more accurately reproduce human physiological conditions than the Vero cells. Because SARS-CoV-2 does not infect mice, so many researchers are using hamster, ferrets, and cynomolgus monkeys. However, it is difficult to use these animals in large number in the infection experiments, so the organoids that can reflect human physiological condition are expected to be an attractive resource. And this table summarizes the organoids used in SARS-CoV-2 research. It has been known that the SARS-CoV-2 can infect bronchi, lung, kidney, liver, and intestine, and blood vessel organoid. And our research team generated bronchial organoid and applied them to SARS-CoV-2 research. And SARS-CoV-2 is known to infect many organs, not just the respiratory tract. So it is speculated that this is one of the causes of multiple organ failure in the COVID-19 patient. Organoids can be used to identify cells and organs that are infected with SARS-CoV-2. SARS-CoV-2 studies using airway, alveoli, and cardiac organoids are well performed. It has been reported that the CiRA cells and club cells are infected with SARS-CoV-2 using airway organoids. In the alveolar organoid, type 1 alveolar epithelial cells are not infected, but type 2 alveolar epithelial cells are infected and the expression level of surfactant proteins is reduced. In the cardiac organoid, the virus does not infect endothelial cells or fibroblasts, but virus can infect cardiomyocyte, causing a diastolic dysfunction. Most of the infection selectivity of SARS-CoV-2 can be explained by the expression of ACE2. So by using organoid, it is possible to identify the cells to be infected, and also predict how the organs will be damaged by the SARS-CoV-2. Organoids are being used to investigate how the viral RNA genome of SARS-CoV-2 is recognized by the host cells. It has been reported that the MDR5 acts as an RNA sensor in the cells infected with SARS-CoV-2. After MDR5 recognizes the viral RNA genome, type 1 interferon is produced. And after that, the gene expression level of many interferon-stimulated genes are increased. Organoids have been also used to analyze proteins required for SARS-CoV-2 infection. And TMPRSS2 is required for SARS-CoV-2 infection in alveolar organoids, but it has been reported that the cardiac organoid requires cathepsin instead of TMPRSS2. These results show that the organoids are also useful tools for elucidating the infection mechanism of SARS-CoV-2. Development and evaluation of COVID-19 therapeutic agents using lung organoids are also being carried out. RDRP inhibitors and inhibitor of receptor and protease have been evaluated by using lung organoids. In addition to evaluation of existing COVID-19 therapeutics, such as remdesivir, new drug development is also being carried out. And Heckman et al. Found several antiviral candidates that cannot be found in Vero cells by screening using organoids. And Hans et al. Also found the promising 3 viral entry inhibitors from a library of FDA-approved drugs. And I hope that the compounds found using lung organoids will give good results also in clinical trials. In addition to lung organoid, cardiac organoids are also used in development and evaluation of COVID-19 therapeutic drugs. As I mentioned earlier, cardiomyocytes use cathepsin instead of TMPRSS2 for priming viral spike protein. Therefore, TMPRSS2 inhibitors, such as camostat, do not block the SARS-CoV-2 infection, but the cathepsin inhibitor, such as ALM, block the SARS-CoV-2 infection. Among type 1, 2, 3 type 1 interferons, only interferon beta has been reported to have antiviral effects in cardiac organoids. Is similar to lung organoid, screening aimed to develop new COVID-19 therapeutic agents is also being conducted using cardiac organoids. And Garcia et al. Have found compounds that have antiviral effect not only against SARS-CoV-2 but also against SARS-CoV-1 and MERS coronavirus. And organoids are considered to be useful tools not only for evaluation of existing COVID-19 drugs but also for development of new COVID-19 drugs. And the introduction was a little bit long, but from here I'd like to introduce the SARS-CoV-2 research in my laboratory. We are now conducting SARS-CoV-2 research using bronchial organoid and iPS cells. And we are also using CRISPR-I system to search for and evaluate genes which are important for SARS-CoV-2 infection and replication. And this is a schematic representation of different areas in the respiratory system. The human respiratory system consists of bronchi, small bronchi, and alveoli. Because the SARS-CoV-2 is first exposed to bronchi among these tissues, so we decided to first generate human bronchial organoids. And bronchi are composed of basal, club, goblet, and ciliated cells, as shown here. And basal cells have property of stem cells, and club and goblet cells are secretory cells. And ciliated cells exist on the respiratory tract side and have many cilia on the cell surface. and we used two models in this study. One is the bronchial organoids, and the other is air-liquid interface model. Organoids are very good model, but they have inside-out structures. In other words, the apical site is on the lumen side of the organoid. To perform virus infection from the apical side, the bronchial organoid-derived air-liquid interface model was used in this study. Therefore, in our research, we conducted SARS-CoV-2 experiment by using these two models. In our study, we generated human bronchial organoid from commercially available human bronchial epithelial cells. Human bronchial organoids can be generated only in 10 days, and they also have high replication potential, so they would be an ideal model for drug screening. We also performed immunostaining analysis in our bronchial organoid. Our organoids were positive for cytokeratin 5 and associated alpha tubulin, suggesting that our organoid include basal stem cells and also ciliated cells. And these results also indicate that the basal stem cells are located outside of the organoid. On the other hand, ciliated cells are located in the lumen side of the organoid. We also obtained 10 images of our bronchial organoids. 9+2 arrangement, cilia, and microvilli could be observed in our organoid. And these 10 images also show that our bronchial organoid have bronchial characteristics. Here we perform SARS-CoV-2 infection experiment using our bronchial organoid. All infection experiments were performed at BS3's facility. After the SARS-CoV-2 infection, intracellular viral genome and progeny virus could be detected. Therefore, it was shown that the SARS-CoV-2 infection could be evaluated in bronchial organoid. However, the production efficiency of the progeny virus was very low. And after 5 days from the SARS-CoV-2 infection, we examined the viral spike protein expression in the infected organoid. The viral spike positive cells were localized in the outer edge of the infected organoid. And this data shows that the most cells were not infected with the virus. And in addition, no infected cells could be observed more than 1 week after the SARS-CoV-2 infection. We uploaded the data of SARS-CoV-2 study using bronchial organoids to bioRxiv in March 2020. After that, our research was introduced in Nature News in June 2020. And since our bronchial organoids do not contain vascular structure or blood flow, so they cannot reproduce the cytokine storm of COVID-19. So we are now currently using organ-on-chip technology to generate bronchial organoids with blood vessel and blood flow. So we hope that this will enable the development of anti-inflammatory drugs by using organoids. As the next topic, I will introduce the SARS-CoV-2 study using undifferentiated iPS cells. There are large individual differences in the symptoms of COVID-19. It is known that about 20% of COVID patients become severely ill. Perhaps the most important cause of the severe COVID-19 is aging, but the difference in genetic background is also thought to be one of the important causes. So iPS cells are a promising tool for investigating the relationship between the genetic background differences and diseases. And human iPS cells can be established from almost any individuals. And at CiRA, iPS cells have been established from over 500 individuals. And by conducting SARS-CoV-2 infection experiments by using iPS cell panel, we may be able to reproduce into individual differences in COVID-19 and also clarify the causes of individual differences. Because it is time-consuming to differentiate somatic cells from undifferentiated iPS cells, so we investigate the possibility of performing SARS-CoV-2 infection experiments by using undifferentiated iPS cells. Here we examine the expression level of viral receptor and proteases in undifferentiated iPS cells. The expression of ACE2 was very low in undifferentiated iPS cells, but that of CD147, which is known as the receptor, was very high in iPS cells. And TMPRSS2, which is a protease, was expressed in undifferentiated iPS cells. We infected undifferentiated iPS cells with SARS-CoV-2 in the BSL-3 facility, but the morphology of the iPS cell colony did not change. And also, no cell expressing nucleocapsid positive were observed. So therefore, this result indicates that the SARS-CoV-2 does not infect undifferentiated iPS cells. Because human ACE2 and TMPRSS2 are known to be important for SARS-CoV-2 infection, human ACE2 and TMPRSS2 were overexpressed in undifferentiated iPS cells. By overexpressing ACE2, a large amount of virus in the iPS cells was detected after the SARS-CoV-2 infection. On the other hand, the effect of TMPRSS2 was limited, and therefore ACE2 expression is required for SARS-CoV-2 to infect human iPS cells. And 2 days after the SARS-CoV-2 infection, cell fusion was observed. 4 days after the SARS-CoV-2 infection, many of the cells died. And 10 images of ACE2 expressing iPS cells infected with SARS-CoV-2 are shown in this slide. Virus particle attempting to invade the cells could be observed as shown here. In addition, viral particle budding in the intermediate between the ER and Golgi, which is known as ER-JIC. Furthermore, the DMV was also observed. DMV is the double-member vesicles, which are the central hubs for the viral RNA synthesis. And these images show that the life cycle of SARS-CoV-2 can be observed in ACE2 expressing iPS cells. Next, we perform immunostaining analysis of ACE2 expressing iPS cells which were infected with SARS-CoV-2. By infecting the cells with SARS-CoV-2, viral and protein positive cells were observed as shown here. Some of the infected cells had fused with each other. And this image was taken 2 days after SARS-CoV-2 infection, but 3 days after the viral infection, all cells expressed viral N protein. And we also perform RNA sequence analysis in the uninfected and infected ACE2 expressing iPS cells. And color dots in the volcano plot indicate the genes whose expression level changed significantly more than 4-fold. We are currently analyzing the function of the genes whose expression level have changed. And we also confirm that the gene expression levels of the undifferentiated markers remain unchanged after the SARS-CoV-2 infection. And next, we examine whether ACE2 expressing iPS cells could be used for drug screening. Here we test the 8 drugs used in the COVID-19 clinical trials, and Vero cells were used as the control cells. And after exposing the cell to various concentrations of the drugs, the number of the viral RNA copy in the cell culture supernatant was quantified. A sigmoid curve was created and EC50 value was calculated. And among 8 drugs, the antiviral effect of remdesivir was the strongest. We also confirmed that the ACE2-expressing iPS cells are more sensitive to remdesivir than the Vero cells. On the other hand, the chloroquine favipiravir, which is known as Avigan, did not exhibit viral replication at all. Did not inhibit viral replication at all. For drugs other than interferon beta, the effect of drugs was more strongly detected in ACE2-expressing iPS cells as compared with the Vero cells. Although we skipped the details, but we were able to confirm the antiviral effect of RDRP, such as remdesivir and EADD-E2H01, and TMPRSS inhibitors, such as camostat and nafamostat, in ACE2-expressing iPS cells. So this result indicates that the ACE2-expressing iPS cells can be used to evaluate COVID drug candidates. So we do not have enough time, so we skip the details. But we also investigated the cytotoxicity of these drugs. And it is also important to confirm the antiviral effect at the concentration that does not cause cytotoxicity. And here we perform SARS-CoV-2 infection experiment using human ES and iPS cells established from 8 donors. The replication efficiency of the virus was different among human ES and iPS cell lines. We confirmed that the viral replication efficiency in male iPS cell was higher than that in female iPS cells. And we examined some gene expression to find out the cause of sex differences. And as a result, we confirmed that the TMPRSS2 is strongly expressed in male iPS cells. However, this point needs to be analyzed in detail. For the above, this result suggests that the human iPS cells can be used to reproduce individual differences in the SARS-CoV-2 infection and replication. However, since S2 is overexpressed in this study, our method is not effective in the cause of individual differences is S2 or its related genes. And this is a summary of SARS-CoV-2 studies using ACE2-expressing iPS cells. We found that the ACE2 expression is required for SARS-CoV-2 to infect undifferentiated iPS cells. And in this study, we used adenovirus vectors to overexpress ACE2 in multiple iPS cell lines. But when we perform infection experiment with single iPS cell line, we use the iPS cells that constitutively overexpress ACE2 by using piggyback vectors. We also confirmed that the COVID-19 drug candidates can be evaluated by using ACE2-expressing iPS cells. And by using human iPS cell panel, individual differences in SARS-CoV-2 infection can be reproduced. And since we use 8 human ES and iPS cell lines in this study, we use a very large amount of stem-fit medium and I matrix. Although many cells are required for COVID-19 drug evaluation, iPS cells cultured in stem-fit medium have a high replication capacity so that sufficient cell number of iPS cells can be easily prepared. Today I talked about 4 topics. We are conducting SARS-CoV-2 research using bronchial organoid and iPS cells. Since the development of therapeutic drugs is behind the development of COVID-19 vaccine, so we believe that the drug discovery research using organoids and iPS cell will become even more important. We are already collaborating with researchers from many countries, and the new collaboration is of course welcome. So we are looking for And we are also looking for postdoc and students to study COVID-19 together. And if you are interested in our work, please feel free to contact us. And I would like to thank the lab members for their hard work, and also I want to thank all of our collaborators. And at last, I would like to thank again AMS Bio, Matrixome, Ajinomoto, and Xenoac for giving me such a great opportunity. And thank you for your attention. I'm happy to take any questions.